Hello, my name is Sean Nario, and this is my submission for mini exam video for Prompt 1. On the top left of the first page, I have attempted to draw the Watson-Crick model of DNA. The image contains a key labeling yellow adenine, pink thymine, green guanine, and red cytosine. The key also has A and G listed as purines, double ring structures, while T and C are listed as pyrimidines and only have one ring. Using this key, we can see that A's bind to T's and G's bind to C's, forming a base pair. These base pairs are stacked closely on top of one another. This is called base stacking and prevents the bases from interacting with water, giving them the hydrophobic property. Though poorly drawn, I have attempted to show the handedness of this DNA structure to be left-handed, indicated by the red arrows here. The major and minor grooves are also shown here as a result of the base pairs separating the phosphate backbones, allowing for more protein or solvent interaction with the bases in the major versus the minor grooves. As seen in the structure, the 5' and 3' ends of the DNA backbones are labeled, matching with the opposite ends, resulting in it being anti-parallel. To the right of the model box are the organic structures of the 4 bases, also labeled with their purine and pyrimidine designations. Here I have defined hydrogen bonding and the elements that are capable of hydrogen bonding. For the purpose of this presentation, we will only be focusing on nitrogen, oxygen, hydrogen bonding, as they are seen to the right of the hydrogen bonding box. The dashed blue lines indicate hydrogen bonding forming the base pair. Keep in mind, hydrogen bonding is not the main force keeping DNA together. The antiparallel phosphate backbones of the DNA strand keep the DNA together by being hydrophilic, attracting water to the backbones instead of the base pairs. Towards the top of this page, I have drawn the same example DNA as seen on the previous page. The box immediately to the right is a zoomed-in look at the bond structure between the phosphate backbone and the nitrogenous bases, with emphasis on the structure of the backbone. As you can see, the backbone is made of alternating sugar and phosphate groups. The negative charge in the phosphate group gives DNA its negative charge. Below the backbone structure bond diagram, I have isolated and labeled a phosphate molecule as well as a deoxyribose sugar molecule. The sugar molecule is labeled with carbons 1 through 5. To the left of the deoxyribose sugar box, I have the significance of carbons 1, 2, 3, and 5 outlined. Carbon 1 bonds to nitrogenous base. Carbon 2 is missing the CO bond, making a deoxy. Carbon 3 bonds to following phosphate group, and carbon 5 bonds to the previous phosphate group. Towards the bottom of the page, I have a simple diagram showing the effect of a SSC, a salt solution buffer, with partially negative DNA. The salt buffer interacts with a negatively charged DNA, partially neutralizing it, which reduces the effects of destabilizing forces. Reducing the effects of destabilizing forces allows DNA to bind more tightly, which increases the TM value, or the value at which 50% of DNA is denatured. This concludes my submission for video exam 4. Thank you. And here are my sources.